Hello, everyone. So my name is Daniel Smith. I'm a postdoc at the University of Arizona in Joanna Mazel's lab. And I'd like to start things off with this quote from Stephen Stearns that I find amusing. Fitness, something everyone understands, but no one can define precisely. Indeed, listening to talks throughout this conference, there's the start bell, I've heard a few talks that do not reference fitness in some way. These references are often shorthand for something, often with respect to a specific model. But how useful is this imprecise use of fitness? And so today I'm going to talk about what work does fitness do, and what can fitness help us understand? So what work does fitness do in population genetics models? And I'm not talking so much about inferences, the sort of what-if questions that many of us ask, but its most basic use in theoretical models. What we do is we assign some kind of short-term fitness to some model from which we derive longer-term outcomes. We do this to answer questions about what evolves over longer-term timescales. For example, what fitness we assign is different in different models. Absolute fitness, the expected number of offspring produced by an organism over its lifetime, is an example of an assigned fitness that we often use in annual plant models. We often say assign a mutant to have fitness 1 plus S. And with this assigned short-term expected fitness of 1 plus S, we might ask how likely is it that this beneficial mutant escapes rarity or fixes in the population. And a classic model might derive that probability. Or similarly, we might assign relative fitness 1 plus S, which depicts the relative contribution to the gene pool of an allele. And like somewhat confusingly, we might use the same S's, even though they are not mathematically the same. But I argue that we're not actually assigning either of these things in principle. What we're in fact doing is not assigning fitness. Behind each assignation of fitness, we are in fact assigning vital rates of birth, death, and growth. For example, take absolute fitness, aka lifetime reproductive success. If you have birth rate B and death rate D, the flower is supposed to be on its treadmill of life, you get lifetime reproductive success, which is trivially derived as B divided by D, births per life. And it's described in terms of vital rates, from which we might derive something like fixation probability. But this is not always an appropriate fitness metric. Many models, particularly those with an ecological flavor, might examine the Malthusian fitness R, which is the difference in birth and death rates, and which does not contain the same information as lifetime reproductive success W. If you have overlapping generations, and sort of trivially, you could have some trade-off between B and D, where you get a genuine mismatch between what these quantities tell you. And furthermore, you might think about the vital rate of growth, and that also would factor into like the speed of evolution, which is the Malthusian parameter. And so, in summary so far, different fitness operations encode different information. Fitness operations such as absolute fitness or Malthusian fitness stem from some kind of assigned vital rate. They're this sort of murky shadow that we derive beneath vital rates. And what do they tell us? And so I'm going to extend our example, thinking about births and survival, which are also affected by temporal variation. And I'm going to talk about this example because I want to show that R and a long-term fitness metric like fixation probability can give strikingly different predictions, and I want to use this to sort of motivate how we might think about fitness in a useful way. So consider an annual plant which experiences good years, in which seeds reproduce upon germination, and fail to reproduce if it's a bad drought year. And staying in a seed bank avoids extinction on bad years. And there are many, many models that consider the evolution of annual germination probability G. One classically calculates the value of G that maximizes the Malthusian parameter averaged across environments, also known as maximizing the geometric mean fitness. But I want to look at this and compare how this value of R might compared to fixation probability of lineages. So this dashed red line shows the evolutionary stable state with a set of parameters of germination probability in a model without demographic stochasticity, such that lineages only experience deterministic extinction. This is what you would get, for example, from an adaptive dynamics model. And these points here show the long-term stationary frequency of germination probability G as derived from fixation probabilities of stochastic lineages with demographic stochasticity. And the simple point I'm making here is that the long-term fate of lineages may differ systematically from what you would expect from the short-term fitness metric. So what's going on here in a model like this? How should we think about these mismatches, and how do we get conceptual clarity when we're trying to think about fitness? And so I'm going to now present how we conceptualize fitness. So when fitness enters models, again, at some point we're assigning vital rates of birth, growth, and death. And in the case of many models, such as the annual seed bank model I just talked about, we are assigning how the specific selective environment phenotype match leads to a particular set of vital rates, and we're deriving short-term fitness metrics such as the Malthusian fitness or absolute fitness over the average environment. And so what we have here again is short-term fitness is this murky shadow underlying vital rates. And next, as we're thinking about fitness, we allow feedbacks. There will be a feedback loop between vital rates and environment. All births, deaths, and growth affect the environment in terms of, for example, population density, frequencies of types, which might be important in game theory models, and things like biomass. And the phenotype also affects the environment. This could be something like migration, or where the environment is chosen by an organism, or it could be something like niche construction. It's some concept of the extended phenotype. And in addition, we have some genotype to phenotype processes going on in our models. In some models, these are ridiculously simple. For example, as in the first example I gave, you can simply collapse everything in the environment and the organismal phases just to a phenotype that gives a particular birth rate. That's like the simplest model you could do. The point is that there is a distinctive assumption about the processes by which a genotype goes to a phenotype type, and through that, from which they become vital rates. And in more complex models, we explicitly considered the developmental environment and informational environment, which I'll describe in a moment. Between the genotype and phenotype map, we have something that we call strategy. So in our view, the strategy in the context of the informational environment is what an organism more or less tries to do, quote unquote, or has a trajectory or developmental commitment towards doing. The informational environment consists of things like signals or cues that an organism can use. That might be something like light availability or quorum sensing, something like that. The phenotype, and then the developmental environment is a conceptualization of how the developmental pathways are actually realized in terms of a phenotype. If there's insufficient resources to develop a certain morphological feature, that phenotype isn't realized. And then ultimately, the phenotype meshes with the selective environment, and that is what provides vital rates. And so what different models do is they make different assumptions at different parts of this scheme, and think about fitness or what evolves in some capacity. And in our view, what we want to know, often when we're asking questions, we are usually asking questions about what kinds of strategies evolve as such. These are the things that are often kind of biologically complex and that we think are interesting. And sort of to close the loop on this figure, what we really want to understand are genetic lineages. Genetic lineages are often framed as, for example, when you introduce a mutation, what happens to it? And our idea of long-term fitness is this sort of hazy shadow of the entire complexity of the system. But specifically, it's a hazy shadow that represents the fate of genetic lineages, which is a sort of complex process that relies on all the feedbacks of these different aspects of our models. And I want to sort of emphasize lineages per se for a moment, because I found it's often a source of confusion. So lineages, in essence, they've been described as the thing that sort of bear fitness, at least in our view, long-term fitness. And in our view, like short-term fitness doesn't really exist as such. They're simply vital rates. A lineage, also to be clear, is a sort of physical object in space-time. It's defined by the physical act of inheritance, stemming from the point at which it appears to the point it either fixes or goes extinct in a population. And sort of adding on this, if you have recombination, each gene's lineages are nested differently within a shared organismal genealogy. This relates to work about ancestral recombination graphs. It's beyond what I want to talk about here. But the point is, things like recombination in sex are not a threat to this lineage-based perspective. This kind of perspective excludes alleles generated by different mutations. Those are, or the same allele generated by a different mutation. Those are different lineages, but it includes things like second-order mutations, and you can ask questions about things that relate to the evolution of evolvability and mutation rate. And also would include things like horizontal gene transfer, which I think kind of interestingly sort of leads to some breakdown of the classic ecological evolutionary divides. So I'm arguing that what we actually care about is long-term fitness, and lineages capture this information. And so how does one summarize this? And there are many ways. One way that's increasingly common in theoretical work is to take the ratio of fixation to counterfixation probabilities in your model, in your stochastic model. This is, it's basically sort of taking the odds of rare symmetric mutation in which you're then asking what states are you most likely to be in due to selection. And you can also add things like mutation bias. Another way is thinking about inclusive fitness. Which you can sort of, some interesting arguments have been made that describe this in terms of lineages rather than individuals, which I find very pleasing. And since I'm running a little bit short of time, I believe, we also derived some lineage-based metrics for looking at balancing selection. This example is with a stochastic version of the Hawk-Dove game. I don't have time to highlight what I wanted to highlight, but just want to say that the main thing I want to present here is this picture. This picture is a way of thinking about and deconstructing fitness, whether it's sort of long-term or short-term. Fitness is this sort of shadowy thing, and different models are simply trying to get at this in different ways, but the hope is that we have tried to go towards some conceptualization that allows us to ask pointed questions about fitness and strategies. There's examples from the literature that sort of try to do this that we think are valiant attempts. And so in closing, we think we need to be precise with our language, what we mean by fitness at different temporal scales, and what questions we're asking. Fitness is sort of these shadowy, murky things, and they're often particularly confusing because the same terms are used without the kind of rigorous definitions that are required. Specifically, I would say we should ditch short-term fitness and just talk about vital rates, and then talk about lineage fates, which is fitness in the long term. So I'll leave it there. I'll say thank you to my talented and wonderful collaborators, and there's maybe time for a question.
Okay, so thank you very much for coming all the way to the farthest end of the conference center to hear me talk about adaptive gene amplification on extrachromosomal circles. Yes, so extrachromosomal circular DNA are extrachromosomal DNA elements. They're also circular. They're commonly found in the nuclei of a large variety of eukaryote cells. And their content is just a copy of a region of the chromosome. So they're not like plasmids that have complex regulatory functions encoded on themselves. They're just a copy of a bit of the chromosome. The mechanism of production is a little bit unclear, but it seems to involve, it seems to happen as a byproduct of either transcription or replication of the chromosomal DNA. And Oh yes, and of course this is, provides an opportunity for a gene amplification, because if the region of chromosomal DNA that's on your extra chromosomal circle has a gene in it, you now have extra copy or copies of the gene, as many extra copies as you have extra ECC DNAs, and this can have important fitness consequences. So the motivating example here is transcription-limited copper resistance. So this is based on some experimental work from the group of Sally Otto, where they were doing evolutionary rescue experiments in yeast and exposing them to toxic concentrations of copper. And in some of their evolved rescued lines, they found that the mechanism of copper resistance was a high copy number ECC DNA that had a metallothionein gene on it. So basically, in order to survive high concentrations of copper, these cells need a lot of metallothionein. And one of the things that, at least, that's preventing them from getting enough metallothionein is producing enough of the mRNA. So having a large number of ECC DNAs with the metallothionein gene on it gives you a significant advantage. And so we're interested in modeling this scenario, and we're interested in sort of answering two big questions. The first is sort of how many extra chromosomal circles should you expect to find in your cells in your yeast population, and what's determining that? So how do you go from sort of the underlying biology of the ECC DNA behavior inside the nucleus to finding out how many ECC DNAs you typically find in cells in ordinary populations, and then in populations where the ECC DNAs are doing something important for these cells. And then we want to know, under what circumstances can these ECC DNAs then help rescue these populations from things like exposure to copper? So when can these ECC DNAs increase the probability of survival for the yeast population? All right. So yes. So the model I have here. Sorry, I can't see my slides on here. The model I have here is we have a population of yeast. And we divide we'll have a lovely stochastic process, and we divide all of our yeast into countably infinitely many types, indexed by the number of ECC DNAs have. So from 0 ECC DNAs through arbitrarily many. And they're going to go through a few different processes that we have to keep track of. The elementary reactions for our stochastic process. So first there's some processes that happen purely to the ECC DNA. So these are the processes within the nucleus of a single cell. And the first of these is just production of new ECC DNA copies. So if you're not familiar with the notation here for elementary reactions of a stochastic process, this is just saying that you can go from a yeast of type N, that is, to a yeast of type N plus 1 at a rate nu. And these events happen at a Poisson process. Right. So this nu is the per capita rate of production of new ECC DNAs from the chromosome through this mechanism that's not fully understood. And there's some possibility that these yeast might be able to be replicated inside the nucleus themselves. So replicating the new ECC DNA copies from existing ones. So we let that happen at a per capita rate lambda. And then the cell might be able to sort of degrade these ECC DNAs to recover nucleotides. And so we might allow this to happen at some per capita rate mu. So that's everything inside the nucleus. And then we need also to keep track of the yeast population itself. So what are the yeast cells themselves doing? And so they are doing two things. They're being born. And so they are born at a birth rate B sub n. Oh, it's all the way on the, I don't have a pointer. Okay. They're born at some rate B sub n, which might depend on the ECC DNA copy number n. That's the selective effect of our ECC DNAs in general. And then the rest of this sort of complicated rate expression is tracking the fact that when a new yeast when a yeast cell divides, we have to keep track of the segregation of the ECC DNA copies between mother and daughter. And so this, we believe this happens sort of just randomly. So that's why we have randomly and independently for each ECC DNA copy. So that's why we have a term from a binomial distribution here. But yeast do a funny thing. So budding yeast, like Saccharomyces, have asymmetric division. So there's a clear mother and daughter cell. And the mother cell can selectively retain lots of sort of junk in itself. And that junk includes, in many cases, ECC DNAs. So we've added a sort of biased random segregation property here where this p parameter measures the degree of bias. So at p equals 0, there's no bias in segregation. This will be ECC DNAs will be perfectly equally likely to go to mother or daughter cell. And then as p increases, they're more and more likely to stay in the mother cell. up to p equals 1, where they're certain. And the last thing that the cells are doing is dying at some rate that, again, might depend on the ECCDNA copy number in our selective case. All right. But for the moment, it's not going to depend on the ECCDNA copy number. So we'll just start in the neutral conditions, where the ECCDNAs aren't actually doing anything helpful for the cell, and we just want to know sort of what number of ECCDNAs should we expect when they're just sort of neutral and uninteresting for the cell population. So in this case, our birth and death rates are just constants. They don't depend on the ECCDNA copy number. And the yeast population dynamics now are boring. They're just a birth-death process. But we can look at the distribution of inside a nucleus of the typical copy number. And so the first thing we can look at here is just the moments of this distribution. So the mean number of ECCDs in a typical yeast cell from our population will have, its behavior will depend on this lovely little lambda minus mu minus b over 2 condition here. So this is the lambda minus mu is sort of the net growth rate inside a single cell of ECCDNA copy of the ECCDNA population inside a single cell, and then we're subtracting half of the birth rate of cells themselves. And if this quantity is non-negative, then we are in a probably non-biological scenario where you accumulate ECCDNAs forever. The mean is going up exponentially. And then if this critical quantity is negative, then we get into the scenario where in the long run we expect to go to a sort of a finite mean a finite mean number of ECCDNAs in a cell. And then we can also look at the second moment, or the second central moment, the variance, and it does a very similar thing, but with a slightly more complicated critical expression here. So now, in addition to, but when this quantity is non-negative, then the variance is increasing without bound. When it's negative, the variance is going to a finite value that won't fit on the slide and isn't particularly edifying anyway. And so the only interesting thing here is, of course, that the degree of bias and segregation doesn't have any impact on sort of the mean field behavior of the ECCDNA distribution, because after bias segregation, both the mother and daughter cell are still contributing to the population, but it does have a notable effect on the behavior of the variance. As you get, unsurprisingly, as you get more and more bias segregation, there's more and more variability between cells in the population. I'm afraid that is the last analytic results that there will be in this presentation, I'm sorry to say. So everything after this will be from simulations. So we can look now at simulations and look in what the long-term stable distribution of ECCDNA copies will look like in it full, not just looking at moments. And it looks like this lovely thing. So this is for two slightly different values of the sort of mean of the distribution. We get, it's a sort of very typical long right-tailed distribution. And then the sort of interesting thing here is, this was unbiased segregation. Now we're adding in also the strongly biased segregation, and we can see that this strong effect of the biased segregation increasing the variability in this distribution, and as a result, also driving us towards increasing the mass on these very low copy number end of the distribution. All right. But we were really interested in cases where the ECCDNA are doing something useful for the cell. So now we have to pick actual functional forms for the vital rates of this ECCDNA of these yeast population. And so for what I will present today, we're just going to keep our birth rate constant, and our death rate will decline hyperbolically here. So the idea is that very low ECCDNA copy numbers, this population is doomed. Its death rate is higher than its birth rate. It is certainly going extinct. And then eventually this breaks below 0, and you get more and more likely that this yeast population will survive. All right. And we can ask again, first, what does the typical number of ECCDNAs look like in a cell in this selective conditions? And this is what it looked like. And I promise I did not make a mistake and accidentally put the same slide on here twice. So if we can look, you can actually compare selective and neutral under these two conditions. And you can see that selection itself doesn't do very much here. I mean, there's a slight increased accumulation of ECCDNA copies. There's a slight shift of this distribution towards the right here. But selection itself, it doesn't affect really, doesn't have a very large effect on the typical number of copies of an ECCDNA that you find in the cell. And then when we look at bias, the connection between the comparison between biased and unbiased segregation has very similar effects under the selective model. In particular, it's bias bias segregation is heavily increasing the mass on these low copy numbers, which now in the selective model these are the doomed cells, right? And so this brings us to the last result, which is the thing that we're really interested in, which is, can this help the yeast population survive? All right, so here's the answer to that question. So on the y-axis here, this survival probability is a single cell survival probability. So you imagine you start off from a single yeast cell and ask, what's the probability that its descendant population will still be around, in theory, indefinitely far in the future, because this is simulation results. So I think it's actually 50 generations far in the future. And so remember, if without ECCDNAs under the fitness function we chose, this probability would just be 0. So the ECCDNAs are doing something helpful here, is the answer. And so along the x-axis, we have a measure of sort of it's this critical value coming back again, but this is just a measure of the typical abundance of ECCDNAs in our cells. And unsurprisingly, if you increase the typical abundance of ECCDNAs in your cell, and the ECCDNAs are doing something helpful in reducing the death rate, you increase the survival probability. That's not surprising. The interesting part here is the effect of the segregation probabilities. So you can see very clearly, as we increase the amount of bias in the segregation, then we get lower and lower probabilities of survival of this population. And this is because again, the wider distribution under the biased segregation model is putting more mass on low ECCDNA copy number cells, or to think of it in a sort of different but equivalent sense, you're just when there's lots of bias in segregation, you're making lots of daughter cells that don't have many ECCDNAs and are therefore in trouble. All right. Yes, so this is to summarize up. So looking again at the typical distributions of ECCDNA copy numbers, so these are, unsurprisingly, these are driven by, the birth, death, and production of ECCDNAs within a single cell. This is not particularly surprising. The more interesting thing here is perhaps the effect of the effect that of the birth rate of the yeast cells themselves heavily dilutes the number of ECCDNA copies, and the effect of variance. And yes, and the ECCDNA copy can, in fact, rescue our yeast population. And this is sort of mediated by the typical abundance and variability between cells in the population of the number of ECCDNAs. All right, I'd like to take the opportunity to thank the other members of my group, the rescue team, and the people who give me money, and all of you for your attention. Thank you.
Hi, everyone. Welcome to my talk. My name is Haoqing. I'm a PhD student from Sebastian Hohner's lab at LMU Munich. Today I'm going to talk about our work on gene family evolution modeling. Although it's still a work in process, we only have some preliminary results, but I think it's really interesting to share with you guys. To start with, gene families are defined as a group of genes descending from common ancestor, and we usually start their evolution by investigating their gene family size variation. Here on the left is an example from the work of Sharpton in 2009, in which he estimated the gene family size in several fungi taxa. And you can see that there is considerable variation, not only in the family size within one gene family across the phylogeny, but also there's different patterns among different gene families. So one strategy to study this evolution is by reconciling gene trees with species trees. Here is on the left is an illustrated example. Basically, what we do is try to estimate the number of gene gain and loss by inferring key events like speciation, duplication, and gene loss. Another strategy would be in a more probabilistic framework. Han in 2005 introduced birth and death process to model gene family size variation along a phylogeny, in which birth is basically a gene is being duplicated and fixed, death is a gene is lost. And two key parameters would be the birth rate and the death rate. Before I show our implementation, actually this kind of model has already been implemented by several tools used in study gene family evolution. One of the most common is this CAFE, in which they simplify the model by assuming equal birth and death rate. and they estimate the global rates for all the gene family you fed to them. But their latest version, Cafe5, they kind of allow the rate variation among families by using gamma distribution categories. So our implementation is in REV-based. So why we want to do that? There are several advantages to implement this model in REV-based. First, it allows very flexible model specification. For example, we can have independent gain and loss rate for each gene family. We can also allow different rates on different branches, although we didn't really explore it in my talk, but there is a possibility. And we can estimate ancestral state for internal nodes. We can also do stochastic character mapping. Let me briefly show you how it implements in REV-based. It's different from what they did in Cafe. We describe the birth and death model of gene family use this continuous time MK process, in which is characteristic by a Q matrix, and the element is calculated using the equation I showed here. I can also show you an example, try to give you a picture how we did that. For instance, if we set lambda to 0.5, mu to 0.4, this Q matrix can be, the first line is you start from gene family size is 0, but once you reach 0, you never come back. So that's why the first line is always 0. And for the we start from the second line, which means you start from ancestral status, gene family size is 1. And then the first elements in this line would be you go from 1 to 0, means you lose 1, the death. So it's 0.4 times the number of ancestral value, so it's 0.4. The third one means you go from 1 to 2, so it's a birth events. You just time 0.5 by 1, so it's 0.5. Then you average each line by asking the elements on the diagonal to be minus of the sum of other elements. And because we are doing this like step by step, so you always the +1 or -1, you never jump from 1 to 3 or any other value larger than 2. And then we calculate the transition probability using this equation. We did a quick simulation and validation if we are doing things right in REV-based. So we use the same set, but asking the ancestral value to be 1, and we set an upper bound to 8, which means you can only reach the as much as 8 copies in one in the gene family. This is because we have to have not infinited matrix to do our inference. And you can see that our REV-based results align perfectly with the theoretical probability. Explain a little bit. Like each curve means that, for instance, this one, you cannot see. Like the left one, it means you end at time t with gene copy number, gene family size is 1. And then 0 and 2. The other probability is very low, so I did not show them. And interestingly, we also try to re-implement what they did in Cafe, but maybe there's a little mistake, so it doesn't fit the theoretical line. But we still need to figure out why is that. And then we want to try to apply our model to some real data. Also, my project is working on the evolution in fireflies, so we decided to maybe try some firefly data. I'm not going to talk too much about what I did, what our work on the molecular data part. Basically, we're just trying to generate 2 input file. One is the gene family counts, and one is a good tree, so you can estimate your gain and loss rate for each gene family. These 2 files are also like commonly required by other gene family evolution tools. So it's pretty common. Here is basically what we have. We have a very beautiful tree, but unfortunately, many of our samples, they're still in process. We didn't finish annotate them. So we only, at last, we can only have like a small subtree with 9 taxa, and also we have the gene count of around 3,700 gene families. So we put them into RevBase and try to see what's the posterior of the gain and loss rate. Here is some preliminary results. So on the x-axis, each gene family is sorted by their ID, but it doesn't really mean anything about the ID. So what is important here is you can see there is a variation in not only gain rate, but also loss rate. But if you calculate the gain rate and loss rate ratio, you will find that most of the gene family, they kind of lies between the area, in the area between 1 and 0.1. Sorry. There are some outliers, but we are not sure if these are some interesting gene families to look into, or it's just because these are due to some failed MCMC runs. Oh, wait. And if we plot them with the histogram, we can see very clearly that all the gene family, they kind of lies on the left of the value 1, so which means they have a larger loss value. We also try to plot the gain rate and plot rates against maybe it's affected by maximum gene counts or the difference within gene families. Why is just? Then we found that for most of the families, there might be a relationship between maximum gene counts with gain and loss rates. I mean, this is reasonable because if you imagine that they have if you have a large gain or loss rates in a stochastic process, you might have chance to reach a very large value. But there are also some outliers that lies on the bottom of each plots, which means that these families, no matter their maximum gene counts or the difference within the gene family, they always have very low gain and loss rates. So these might be some interesting gene families to look into. But we are not sure if such pattern is because it's kind of clade specific for fireflies, or because we are only sample like 9 taxa, which is a really small tree. So we tried our model with 2 empirical data sets. One is snail, which has around 25 taxa. One is ants, which have around 80 taxa. And we did similar things. We just put them into our model and plot out the gain and loss rate for the first 1,000 genes. And here is the plots for the snails. You can see for snails, they have much more variation in their ratio compared to fireflies. But when you plot them in histogram, you can find that actually most of them still lie on the left of the line of value 1. And there is a very small peak, tiny peak on the right, which means they have some families which are maybe in expansion. They have higher gain rates. And here is for ants. The ants is more like what we have in fireflies. So most of the gene family, they have a higher loss ratio, although there is considerable variation in their gain and loss rate, but the ratio is always like loss rate is a little bit higher. And what is most important here is that no matter in fireflies or in snails or in ants, you find that like only very little part of the gene family that lies on the line of 1, which means they have equal rate of gain and loss rates. So, which means the assumption in cafe might be interesting to think of that if you really want to assume you have an equal gain and loss rate. So to quickly conclude, we implement birth and death rate in RefBase, which allow us to have more flexible model specification, and our preliminary rate shows a considerable rate variation among families. We found that maybe it is more reasonable to assume unequal gain and loss rates for gene family evolution. And maybe for fireflies, most of the families, they often have higher loss rates, which means most of families, they're in a contraction. I want to thank to my supervisor, Dr. Sebastian Hona and Dr. Anna Catalan. And I also want to thank my colleagues in Hona's lab. And thank you guys for listening. I'm happy to answer questions.